Thank you for the introduction. Thank you all for joining this talk. The motivation of this talk came from my perspective that existing materials in the literature are insufficient to help beginners in setting up clinical trial simulation. There are some helpful papers that describe the concept of simulation study and the underlying details, but they are not specifically tailored for clinical trial designs. Hence, I propose to write a tutorial paper on this topic. I collaborated with the members of the MRC, NIHR, TMRP Adaptive Design Working Group. We wrote a tutorial publication on the topic of clinical trial simulation. We hope that it will help trial statisticians in planning their trial design prior to implementation. This slide outlines the topics that I will cover in this talk. I will first describe the idea of simulation in the context of clinical trials. I will then focus on the OCTAVE framework that we proposed. The elements of the framework suggest what you should consider when planning a clinical trial simulation. I will then briefly talk about some computational aspects. This includes the use of pseudocode, data generation approaches, suggestions on code validation, and approximation methods that may speed up the investigation process. I will end my talk with some comments on graphical presentation and tools. This slide shows other topics that we have covered in our tutorial paper. I'm not going to talk about them here. The first one is related to some statistical aspects used in complex trial designs, while the rest of the topics are related to practice. From my perspective, each topic should be readable on its own without relying on the rest of the paper. You might need to refer to the terminology sections occasionally, as we have used some terms when we write the presentations. So, what is simulation? According to this publication, a simulation study uses a computer program to learn about different situations to understand what might happen instead of doing a real-world experiment. In the context of clinical trials, we can use computer models to explore trial scenarios and outcomes without running the real trial for one or more design and analysis. For example, one trial replication of a simulation study can offer a preview of trial procedures and results before real-world implementation. Consider this simplified example. We have participants who are randomized to study arms A, B, C after enrollment. They were then given the allocated intervention and follow up at a few time points for taking measurements on some outcome variables. So we will have their data. Eventually, we can apply the chosen analysis method to the data and make inference about the interventions. So, in a simulation study setting, we want to mimic the whole trial process on a computer. We can define a data generating mechanism that gives us a simulated data set. Then we can apply the chosen analysis method to get the inference about the intervention from this one trial replication on the computer. If we repeat the process of simulating and analyzing the artificial data m times, we will get m data analysis output on our computer. We can evaluate the properties of a design and analysis strategy by summarizing the outputs over the m replications. For example, for a given data generating mechanism, we can learn the rejection rate of a hypothesis. We can learn if the estimated treatment effect from the chosen analysis method is unbiased. We can learn all this prior to the implementation of a real study by just conducting a simulation. If we repeat the whole process of simulation with different data generating mechanisms, we can actually identify the optimal design setup. For example, we can select the optimal design that has adequate power to detect a treatment effect with minimal cost. These are just some examples of what simulation can potentially lead to. The idea of simulation sounds simple, but the devil is in the details. I believe many of us here have heard of additive designs, platform trials, basket trials, and so on. The motivation of these complex, innovative designs may not be difficult to follow, but we know that their setup details and operating characteristics are not easy to establish. Why? Well, it is because of the attractive and unconventional design features considered in this design. We don't have closed form mathematical expressions to compute what we need. For example, we don't know how to compute the required sample size, say using a calculator or theta function. We don't know what analysis methods can provide valid inference, such as unbiased estimated treatment effect. It is in such context that clinical trial simulation becomes a valuable tool. Specifically, clinical trial simulation can identify trial setups that have design adaptation rules for guiding trial activities or complex statistical rules for making inference, such as the use of Bayesian criteria. Clinical trial simulation can also investigate properties of trial design that test multiple interventions on multiple subgroups before implementation. All these investigations depend on how we define the data generating mechanism. To my knowledge, there's no guidance on how to define the data generating mechanism, including the highly cited ADAM framework. One goal of our paper is to help trialists in thinking through the details of the data generating mechanism for their clinical study. So we propose a framework called OCTAVE for planning the elements of clinical trial simulation. O stands for objectives of conducting a clinical trial simulation. C stands for characteristics of underlying factors. T stands for trial setup. A stands for analysis methods. V stands for valuation, which includes evaluating the property of a trial design and the chosen analysis strategy. E stands for evidence, where we suggest doing so by reporting and ensuring reproducibility. I will now go through the elements of this framework with you. Hopefully, after this session, you know what details are needed when planning a clinical trial simulation for your trial design. In my experience, it is relatively more challenging to define the data generating mechanism for investigation than the other elements. Recall that the data generating mechanism is the instrument that gives us artificial data for the investigation. It consists of the underlying factor and trial setup. Before we start doing anything, defining clear objectives helps us focus on concrete and actionable steps. So our framework starts with O for objectives rather than A for aim. We define simulation objectives by three components in clinical trial context: the underlying factor, trial setup, and analysis methods. Specific objectives are defined by the fixed or varying details of these components. For example, for a single design and analysis strategy, clinical trial simulation can be conducted to assess their sensitivity with respect to some assumptions. One paper has used clinical trial simulation to evaluate the properties of a four-arm, two-stage design with a sequential t-test, assuming different delay lengths in the endpoint. The authors show that the efficiency of implementing a MUMS design decreases when there's a delay in observing the endpoint for making interim decision. Another example of simulation objective is to compare the property of multiple designs that use the same analysis methods under a given set of assumptions. The paper cited here that investigated the performance of different randomization approaches in a platform trial with beta binomial outcome model. If you check the limitation discussed in the work, you will then find the assumptions that they have made throughout the simulation study. So, what is an underlying factor? We define an underlying factor as a variable that is related to trial design and/or conduct. Its true properties or characteristics are unknown to the investigators at the planning stage, and the properties or characteristics cannot be altered by human intervention. Some examples are treatment effect of endpoints, variability in how participants respond to a treatment, prognostic covariates, disease progression, and missing data types. In practice, investigators can select the factors to include in the study and clinical trial simulation, but they hardly know the true numerical representations. For example, they can choose the number of days alive and out of hospital as the primary endpoint instead of a mortality endpoint. But in reality, no one knows the true distribution or characteristic of either endpoint prior to running a study. What we can do is to make assumptions on the characteristic of the underlying variables and then conduct simulation to test the sensitivity to these assumptions as part of the preliminary investigation or site investigation. The next element of our framework is trial setup. A trial design is a structured plan that describes how a study will be conducted to address clinical research questions. In our OCTAVE framework, we refer to trial setup as the statistical and numerical aspects of the plan. These aspects can be selected accordingly by stakeholders. It is something that we have more control than the characteristics of the underlying factors. For example, stakeholders can decide the trial setup based on existing scientific evidence and available resources. They may choose the number of interventions to test at a time according to monetary budget, time, and enrollment rate. The items of trial setup that I've listed here is not an exhaustive list. Out of the many items, the rules for guiding trial activities and statistical decisions are the most challenging one. In my opinion, trial statisticians need to have a rough idea on what the rules can potentially lead to when advising what rules to use for guiding trial activities and statistical decisions. It is this rule that make a design complex. Technically, given the details of the underlying factors and trial setup, we can define the structure and elements of a data generating mechanism. Once the numerical input is specified, we are ready to simulate artificial data for exploration. In our tutorial paper, we define a test case as one that comprises the underlying factors, the trial setup, and specification of analysis model or methods, each set to concrete numerical inputs for this scenario. Some researchers use the term simulation scenarios to describe a numerical specification of the data generating mechanism. But in our framework, our test case includes the analysis model or methods component, as some analysis approaches rely on some user-specific inputs, such as multiple imputation, where we need to specify how many times that we want to impute the missing data repeatedly. Once a test case is specified, we can simulate artificial data, apply the chosen analysis method, and study the replicated data analysis outputs conditionally given the specified ancillary configurations. If one wishes, they may average the findings across alternative configurations, but I do not always find this useful to know. The chosen analysis methods in the simulation study should be the one that would be used in the real trial. We define analysis methods as statistical techniques and procedures used to evaluate accumulating trial data. We know that all analysis methods rely on some underlying assumptions regarding the data or the data generating process. One may conduct simulation study to assess the sensitivity to the underlying assumption of the analysis methods. The summary measures of the replicated inferential outputs inform the properties of the trial design and analysis method. I noticed that many researchers use the term operating characteristic when describing the property of a trial design, and then others would use performance measure to describe the properties of an analysis method. Some commonly considered measures include hypothesis rejection rate, expected sample size, expected trial duration, bias, and mean square error of estimator, and so on. Since clinical trial simulation give empirical values to this measure, good practice should report the Monte Carlo simulation error to capture the variability in the point estimates of these measures. To select a design and analysis strategy for implementation, we suggest selecting a few performance measures to consider. One may have primary and secondary metrics to show what the design strategy can potentially lead to. For example, when considering whether it is worth implementing a particular design, the primary matrix can be predictive probability of success and expected sample size, while the secondary matrix can be frequentist type 1 error rate and mean square error of estimated treatment effects. In addition to the statistical properties, investigators may also consider aspects related to implementation when doing the evaluation of the complex design. It will show a clearer picture as we know the benefits of complex trial designs may not always outweigh the operational and analytical complexity they introduce. We recommend quantifying non-statistical aspects for the assessment whenever possible. Some examples of quantitative measures are listed here for operational complexity, regulatory uncertainty, and the lack of familiarity with novel designs. Lastly, our OCTAVE framework suggests documenting all the aforementioned elements before implementation of simulation for transparency. Clear documentation should explicitly state which components vary in the simulation study. A good report can facilitate reproduction of the simulation results without relying on the code of the author. Record the output of single runs and repeated runs prior to running the simulation on full scale can confirm inclusion of the required inferential analysis and summary measures that reflect the properties. Using this simulation output can also help start the discussion and clarify what is happening with stakeholders. So I hope you now see how the OCTAVE framework structure the details of clinical trial simulation. When drafting the simulation study plan, and before we start writing the computing code, it might be useful to use pseudocode in the process. Pseudocode is a simple, informal, and language-independent way to describe the steps of an algorithm. It breaks the simulation process down into specific steps and decision-making algorithms. It also shows the overall coding structure and flow. Writing the pseudocode helps us identify logical flaws or missing decision points before coding. The figure on the right is an example of pseudocode that I don't expect you to go through. It is also available in our publication. I tend to start with the innermost line, what would happen in a single trial replication. I normally consider the use of the if-else function independently. Then only join the pieces together and moving on to considering the outer loop of what I want to achieve. Another thing that you might want to consider before writing the code is about the data generation approach. One way is to simulate individual participant data. We can generate the data one after another in a dynamic way that emulates real practice. For example, we simulate the baseline data, we randomize the subject, and then generate their outcome data. Alternatively, we can create a database for each trial arm in advance of randomization and data analysis. This is following the idea of potential outcome framework in the context of causal inference. With either way, this approach will add complexity to code and computing storage. Nevertheless, this approach will capture the underlying correlation structures inherently. An alternative data generation approach is to simulate the outcome data at the aggregated level. This relies on the distribution of the aggregated data. That is, we make explicit assumptions on the data. Even though this approach can simplify the code, may be quicker to run and use less computing storage, we cannot explore aspects related to the individual participants. For example, the distribution of the covariates. The correlation between key statistics also needs to be captured correctly when we specify the distribution. Now, I want to comment on code validation. As mentioned, the pseudocode helps break the simulation code into multiple parts. After writing the simulation code, we can first do unit testing to ensure each part functions correctly. For example, we test the randomization and stopping rules in isolation. In between steps, we may include checks that stop the code and provide informative error messages. For statistical analysis validation, we can compare the analysis output to trusted software or known results if using existing methods. For new analysis methods, we may hand-check the simulation results on small data sets or even the largest that we can get, or compare with similar approaches to see if analysis results are consistent. We may also want to test extreme cases to assess the robustness of the algorithm to variations in input parameters. Once we are happy with the data generating functions, we should inspect the simulated data. For example, we can check if the data reflects the intended treatment effects and trial parameters, whether interim analysis data sets are correctly extracted, and trial progression reflects interim decisions. Lastly, we want to do an integration test to ensure that the flow is correct. For example, we first generate individual participant data. We do randomization, then we generate outcome, then apply analysis methods, and make decisions following the rules. After that, we want to have repetitions of the whole process for the computation of performance measures. So what I've described so far may sound straightforward. However, in practice, it can take several iterations before we are ready to run the actual simulation. One challenge that beginners may face when running newly written simulation code is related to computing resources. The code may have passed the validation phase that I've described, but for new beginners, the function that we have written may take a long while to run because we may not know which functions would run more efficiently. So it can take several rounds to update our function and to use a more efficient function to replace the original one. So not only that, when the simulation project has many test cases and each requires a large number of replications, computing resources can become limited. In methodology research, there are proposals of methods that approximate the simulation outputs instead of running all the possible test cases. For example, the ROSA approach and the ADAPT-R package use Gaussian process to model the simulation output, which then provide approximation to the output of test cases that were not evaluated by simulation. The ROSA approach aims to choose an optimal set of test cases for sensitivity analysis with respect to the underlying assumptions, while the ADAPT-R package compute Bayesian multi-arm multi-stage design with early stopping. Another R package, GSB design, they use linear interpolation to approximate the operating characteristic of group sequential Bayesian designs over a range of true treatment effects. These are just some of the proposals that we have mentioned in our publication. I'm sure there are more proposals coming out in the literature. And in our tutorial paper, we are not suggesting that we should use shortcuts like these methods when working on real trial design for implementation. We may be able to consider these tools in the preliminary investigations to identify the exact test cases that need the actual runs, just like the proposal of the ROSA approach. Now, I want to comment on graphical presentation and tools. They are based on our experience. There's no right or wrong way of doing things. When using 2D plots, we suggest plotting the varying component of clinical trial simulation on the x-axis and the performance measures on the y-axis. So I learned it the hard way during my PhD, where the external examiner commented on this. We think line curves connecting discrete points should be used cautiously. In particular, for categorical x-axis, the trend may not be meaningful. For continuous x-axis, dots should indicate the simulated outcome, and readers should be reminded that the line connecting two dots represent approximation. Truncation of the y-axis range may be employed to clarify trends if needed, with all truncated values explicitly reported in the figure caption. We also suggest to include confidence interval computed using the Monte Carlo error when appropriate. Otherwise, we may report the error range in the figure caption. For investigation that examine many varying components, it can be hard to identify trends in the data. Modern tools have been proposed for automated visualizations, which may enable results to be displayed in a multi-dimensional view for identification of unique results. I myself have not used these modern tools, but I believe in either case, it will take several attempts to get the desirable plots or presentations. So I will stop here and happy to take any questions if there's any. Otherwise, I'm happy to hear your experience on clinical trial simulation or challenges that you have faced in your workplace. Thank you.